animals. We will going to study in our medical biotechnology class about the genetically engineered animal. The more famous name is the transgenes. That might be the name which you might wondering. Transgenes. They are the organism with an extra gene or with some additional gene or with a gene of our own choice, with an additional copy of the gene of our own choice. So commonly people thought they are transgenes, but a bigger definition is we will going to study about the genetically engineered animals, and one of the genetically engineered animals are transgenes. We will see why we were trying to create genetically modified organisms or genetically engineered organisms. How many ways are out there to create the genetically modified organisms or genetically engineered organisms. I'm using the word modified organisms because it's more general, but specifically we will be studying only the genetical engineering techniques to create genetically modified animals. Okay, but the similar sort of techniques are applicable to create GMOs, especially the plants, genetically modified plants and other organisms as well. But first and the foremost, why we have to? I mean, this is quite insane of changing the genome of an organism. Why we are interested in affecting or in rewriting the genome of an organism? Why we want to make a change in the genome of an organism? But before going to this to answer this question, when evolution choose to change the DNA, and you know evolution is the master of that technique, changing, creating mutations in the gene, creating different type of DNA from one single copy or from one single organism. So one organism will going to have a different phenotype based on the nature and type of mutations. But evolution took too much time in making and in creating those small little changes. Who knows this will going to survive? I don't know. Let's suppose this one failed to survive, this one failed to survive, and this organism has a mutation which is suitable to the environment and suitable for the very much survival of that organism under those conditions. So that organism live, exist, survive, and thrive, and going to create the entire species. So from here to here, evolution took 100,000 years, so many years to make or to create those small changes and to create a better species than before. Remember, having mutation on the genetic material is a random phenomena, and it depends on probability. There is no law, there is no principle which define that how many mutations will be there and how many of those mutations will be favorable and how many of those mutations will not be favorable. There is no law about all these things. All you need to do is just to think that this is a random probability phenomena. So that is why in several thousand years you develop certain mutations. In the next few thousand years, some of the mutations survived because the organism survived. Some of the mutations were lost because the organisms were lost, because the mutations are not suitable according to their system, according to their their surrounding. So that is how over the period of many centuries, and many centuries is a lesser word, million years, the organisms evolved themselves based on the mutation. They become better and better. Better is not a good word to use. They are evolved. So evolved means the organisms which are more in sync with their surrounding. So since the surrounding is changed, so we need to change ourselves as well. So that the thing which I want you to understand, that this is natural. This is natural to become different. This is natural to become having new properties. This is natural. This is like nature used that thing. But the time span which is required by nature is too much to develop a new feature and a new property. I can tell you it's about millions of years. Now, what we are doing in the last for the last 3 or 4 decades, at least for the couple of decades, what we are doing, we have evolved ourselves in such a way as a species that now we can do this thing. Now we can do the same thing, like one organism, and creating a favorable mutation, a mutation of our own choice, like in a day or so. So now we can do the same thing in days. Now think about, compare the 2 course and 2 pathways. The natural pathway of creating a favorable mutation to change the species took like millions of years. And humans have discovered or developed methods to create the favorable mutations or the mutations of our own choice in a species like in days. So that is, I must say, I want you to grab that concept of the difference in our capacity, what we are doing or what we are capable of. I mean, we are like, I don't know it's hurting the nature or not, but we have developed a technology which has, I must say, outclassed the evolution, because the time factor is the limiting factor in terms of evolution. That is why people will never be able to experience the difference in a certain time span. For the last 2000 years, we might not be able to see any difference in human structures. In evolutionary perspective, it's like nothing. Few thousand years, nothing. But humans now, they have developed the skills to make changes in days instead of millions of years, to experience the evolved version of humans. I don't know that if that will going to be evolved version or not. So the newer version of humans in their lifespans. So we might be able to see the difference during our lifespans. It's not about the millions of years anymore. It's about like few years that we might be able to see an evolved version of species with the favorable mutations. If you ask me why we are doing this, I mean, why we have developed that skill, why humans stay on this path, and why we have masters ourselves for this technology. If you ask me why this path, why humans, because at that time earlier we don't know how the life was written. Now we know that life was written with the 4 alphabets on the DNA, and the sequence defines the difference in different versions of life. We know a lot of molecular biology now. So the next question is that the first question was to discover how life was written. Discovery of DNA. That's like a milestone in human intellectual evolution. I believe that the next milestone or another milestone is the discovery of artificial intelligence. So for me, these 2 are very big milestones of human evolution. So when we discovered DNA, we actually discovered that how the life is written, how nature, writes the life. And you know better than me that the life was written with the help of just 4 alphabets, the G, C, A, and T. Just 4 alphabets. Imagine the level of complexity of life if nature decided to choose 8 alphabets, 20 alphabets, 100 alphabets, 1000 alphabets. You can imagine the complexity of life if you increase like a few more alphabets in writing the codes of life. So that is why I believe that if there is a life exists out there, like an alien version of life is out there, and that the alien version of life is carrying more than 4 alphabets, you can guess how many new functions, how many more functions that life form will be able to execute as compared to us. So the Superman is literally theoretically possible because of they might be having more, alphabets. So that's the first agenda of human race, to know the very much reason of their life. The second thing is that, I mean, once you know about certain thing, once you know about how life was written, what will be the second plan of action? I'm talking about human race. I'm not talking about one scientist or two scientists. क्या होगा? Can you answer me? अगर आप लोग मेरे साथ हैं, अल्लाह करे आप लोग याद हो. महा बोली, महा शाबाश, thank you महा. क्या होगा next plan?
To recreate it.
Wow, shabash, interesting. Anybody else wants to use some phrase?
To manipulate it.
Manipulate it.
Changes.
Ah, shabash.
How to understand it.
Good. So the next thing is that if we know how the life was written, the next thing, how we can write or recreate, manipulate or rewrite the life in our own way, in a way I want to. So if when nature writes the life, nature creates me in this way: 2 ears, nose, eyes, forehead, ear, something like that. If I know the codes of writing the life, I might be able to manipulate. Manipulate is somehow, I like to use this word, but you can choose. We would like to recreate that thing. Customize. That's the better word. We like to customize. I mean, to make changes in our own way. We like to customize the life, to create life of our own, and we have done it. I mean, we have done it. I don't know about the humans. Nothing much was done in terms of human, but think about the other species. हमने chicken को change कर दिया. We have changed the chicken. We have customized the chicken so that it can produce lots of meat to help the survival of the 8 billion human population. We have customized the life in crops and other genetically modified organisms. We are wonderful. The next level is, you can see, customizing life of humans. And I told you, maybe if you were my student one way or another, that the customizing life, according to me, means is to live forever, is to find immortality. So that is the one thing evolution is failed to address. Evolution failed to address, failed to deal with death. It was so eminent, it was so real, it was always inevitable. Evolution failed to find any way how to avoid the death. And I think evolution helped humans to develop that wisdom, to understand DNA, molecular biology, how to manipulate DNA, and to help humans to live forever, together with artificial intelligence. जी, जुनैरा ने hand raise किया था, अब नहीं किया हुआ, पहले किया था.
Sir, can I ask the question?
Sure, why not?
Sir, if we think about it, evolution did find a way to live forever, because evolution happens at the molecular level, and before an organism dies, it does transfer its genes to the next generation. So the genes to survive कर जाते हैं.
Yes, मैंने ये argument दिया हुआ पहले. मैंने reproduction में दिया है multiple times कि death जो है उसको avoid करने के लिए evolution ने एक तरीका ढूंढा. The evolution finds a way, and that is that how about before the synchronous system is gone, how about passing that information to the next generation? There are certain, I don't know, ये कैसा लगता है बोलना? It's weird. Please refer to my video. I have created the video for that thing, that the evolution find that way. But अगर आप देखें, it's not a way, it's a cheat. एक मैंने उर्दू में एक छोटी सी वीडियो बनाई थी, इर्तिका और मौत का मुकाबला. It's about evolution versus death. Evolution और death का मुकाबला. तो evolution के पास कोई direct option नहीं था, तो evolution ने cheat किया. Before the system is disrupted, how about transferring that information to the next generation, and that what we call as the reproduction. और I believe कि ये जो life का जो ultimate achievement है, if you ask me what is the ultimate thing life wants to have, and that ultimate stuff is to live forever. Molecular level पे इसका मतलब ये होता है that life is a synchronous balance between different chemical reactions, and life wants to keep that balance forever. Before that, we found a cheat, and that is the reproduction. But that is not like taking one chemical reaction synchronized system forever. So what we are heading towards now, with the help of our understanding about life and how we can control the life. I can tell you, we understand how life was written, but we are yet not able to understand fully how that life is controlled and regulated. That is more or less like how life, how these genes are regulated. And once we develop our understanding more and more and more and more, that means we will be able to manipulate that information more and more, better and better. And then there will be a time when we might be able to write the life in our own way. And together with artificial intelligence, I believe we might be able to extend the life like for a very longer period of time. Okay, so we are, I mean, अगर आप bacteria के perspective में देखें, in the perspective of bacteria, humans are immortal, है ना? Bacteria तो थोड़े minutes के बाद मर जाता है. I mean, it have to divide, and it has to be converted into newer version, daughter cells. But in their perspective, we are like immortals. We are like existing forever. But in our perspective, we are dying like after 60 to 70 years. We are trying to extend our lifespan. Let's see how it thinks. These are just understanding. So the original debate and the thought is to understand why there is a need to change in the genetics of an organism. Why there is a need. For our scientific and biological understanding, it is required to understand the function of the gene. The very much life we know is revolve around this fundamental thing, that the DNA creates RNA, and that RNA creates the proteins, and the proteins execute the function of life. This is what we call as the central dogma of life. Central dogma of life. So if we have one particular portion of DNA, it might be able to execute one function using the above principle, that part of the DNA will going to produce an RNA, that will going to produce a protein, and that will going to execute a function. So one portion of the DNA will going to produce one function. The second portion of the DNA will going to produce a second function, gene 2. The third portion of the DNA will going to produce a third function. The picture seems to be quite simple and quite understandable as well, that if there are different genes, there will be different function. Let me have some free space from somewhere. Okay, I will be using this. Okay, I'm using this slide to use the free space here. So we have one gene, we have one function. We have another gene, we have another function. We have hundreds of genes, and we have hundreds of functions. And story seems to be quite simpler and understandable. But life is not that much a simple thing to understand. The circuits of life are not connected like this in that much simpler way. So there are other genes, gene 4, which is related to a function 4. There is another thing called G5, another gene, which is related to another function called function 5. So these different functions are connected with each other, because who knows that F2 and F5 will be working together to execute another function called as function number 6. Who knows that the function number 1 and function number 3, they are working together to create another function called the function number 7. If you try to dig and mine that information, you'll came to know that the different genes present on different parts of our chromosomes and DNA, they are not only executing different function indigenously, but they are connected with each other like very complicated circuits, like the circuits in the chip of a computer. That makes very difficult for us to understand the function of that one gene. So if you understand the function of a gene, initially we believe there is a gene, there is a function. So if we want to understand the gene, if we want to understand the function of a gene, we need to delete that gene and to check the function. So deleting the gene will result in the loss of the function of that gene. And that is how we might be able to understand the function easily. The picture was not that much simple. The picture is not that much simple, and it is quite complicated because genes are executing functions not only indigenously like this, but they are executing functions by connecting with each other as well. And that gives you a very complex version of life. So it is very difficult to understand the function of one particular gene. If one gene is involved only in one function, then it's a very simple story. Deleting that gene will give you a different phenotype, and that different phenotype will help you to understand the function of the gene. Adding that gene will give you a different phenotype, and that phenotype will help you to understand the function of that gene. So traditionally, there are two ways to understand the function of a gene. Traditionally, there are two ways to understand the functions of a gene. The one approach is called as loss of function studies. If you will going to enter into this world of discovering and understanding, it's called basic sciences. It's very important, very critical, and very well respected. The other type of research is called as commercial research or applied version of research. If you are in basic form of research and life sciences, you will be studying different functions or a function of different genes. The one of the way is to see the function of the gene by deleting it. That is called loss of function studies. The second way of understanding the function is to add that gene, and that is called as gain of function studies. So these are the two ways people are using right now to understand the function of that gene. By deleting that gene to see what happens, or by giving that gene to see what happens. So that whatever the new things will be happening after that, those new things will be, you can relate those things with the function of the gene. And if you understand this picture, I don't know, I mean, let me praise myself. This is important. If you understand that picture, you'll come to know that if you remove one particular gene, and that one gene is connected with multiple genes within that organism, so the deletion of that one gene will going to affect multiple functions. And that might be confusing, and that might be misleading for the researcher and for the scientist. So deleting one gene in a cell means different. Deleting one gene in an organism means entirely different thing. The ultimate way of studying the function of the gene is to delete that gene at the organism level. That is very important. But that is how you're able to know that how one gene is connected with that many functions. Well, because in an organism, that gene will going to touch other things as well. Similarly, in case of gain of function, you need to add that gene at the organism level, and then you might be able to understand the function of the gene. So originally, creating the genetically modified organism had a theme, and that theme was to study the function of a certain gene. So that is why the people have created the transgenes, genetically modified organism with an additional piece of DNA, with an external piece of DNA integrated into their genome. So large number of genetically modified organisms have been created: mice, cows, pigs, sheep, goats, fish, frogs, insects, and many more, which were unlisted here. Many more which we don't know. Many of the villains out there are creating many organisms by sending any pieces of DNA of their own choices. But remember, still, it is not something which is completely understood. Even we are able to create transgenes, but we don't know exactly how that newer phenotype is actually related to the incorporation of the gene of our choice, because we have incorporated a gene and we don't know it is associated with the phenotype directly or through how many genes. We are still, working and studying. That is why it is very rare that an animal product, no is a strong word now. Please review the literature. I believe there are some products which are coming from the transgenes. Transgenes mean the organism which are carrying an external piece of DNA. It's not easy, not normal to approve those products at FDA or the other levels to be allowed by a human population to consume. Transgenes are allowed, many genetically modified crops are allowed, but any animal which have been modified genetically to produce certain things, to produce certain products, I will show you how many products are there and how we are using them. जी अभी आपने क्वेश्चन पूछना है, हैंड रेज किया है.
Sir, what about broiler chickens? Are they genetically modified?
Please speak again.
What about broiler chickens? Are they genetically modified?
It's genetically modified. Yes. हम उनके दुख दर्द की कहानी बयान करेंगे. ब्रॉयलर चिकन मैं करता रहूंगा उनके बारे में, मुझे पता नहीं क्यों, लेकिन हमें ये समझना होगा. आई एम नॉट द पर्सन who wants to take on the debate whether we have a genetically modified organism or not. This is not the right, because I believe we do need. Without that thing, we might not be able to survive. 8 billion population को survive करने के लिए you need abnormal things, and humans are doing all those abnormal things for the survival of our species. But that ये genetically modified है broiler. So the original purpose is to research. Transgenes का original purpose क्या था? That was to research, to study the function of the gene, to help humans to avoid certain diseases, and to improve the livestock animals, cash crops को बढ़ाने के लिए, and use of animals as bioreactors. That's little bit unkind. That is little bit unkind. Using life and another organism to produce the things of your own choice. So how transgenes are created? How it is possible to create a transgene? Things seems to be quite simple on paper and in the slide, but for the execution of the same thing, you need wonderful expertise. You need a wonderful pair of hands as a researcher to handle the sensitivities associated with such protocols. And I can tell you, you came to know about the trouble which you have done 6 weeks ago or 5 weeks ago. It was just crazy. And you came to know that 4 weeks ago you have put that stuff in that way which you were not supposed to, and you are crying today about the mistake you have done like 6 weeks ago or 4 weeks ago. Research में life sciences के साथ ये बड़ा गड़बड़ है. So how simply we can narrate that thing, that how we can create a transgene with a simple experiment. Okay, have been created. Okay, I will study. So the most common technique which was used to create transgenes were microinjections. If you want to build an organism, and you want every single cell of that organism, is made up of or receives the changes. Every single cell of that organism receives that changes. If you want that, you have to make or create these manipulation at the single cell stage. You have to create those you have to make all those changes at a single cell stage. The single cell stage, or I must say at the zygote level. Naturally, when the sperms and eggs they combine, you can do this experiment in vivo and in vitro as well, and you extract the zygote out of the body of the female, and you send the gene of interest. Now that is a very important question. I'm not addressing this question here. We have indirectly already understand that thing, that how we can send the gene of interest perfectly here. I'm just helping you to recall retroviruses and other plasmids we can use to send the gene of our interest. So how are we able to send the gene of our interest? The first thing we should do is to select a single cell stage. So whatever will going to happen here with that one particular cell, it will going to be part of every cell of the organism. You might be able to succeed, you might not be able to succeed, but if you're able to succeed in sending the gene of interest right within the genome of that organism, that will going to be part of every single cell of that organism in future generations. So the success of the process depends on our ability to send the gene of interest to that single cell. And I can tell you the percentage of success here is less than 10%. Is less than 10%. So like if you are injecting the gene of interest into the zygote of 100 samples, you might be able to get successful incorporation of the gene of interest into the host genome, like in less than 10%. And you never be able to know about the result now. You will never be able to know whether you're able to send, whether you are successful in sending the gene of interest into the genome. You will never be able to know at this. When you will come to know, when that zygote will develop into a mouse, and that mouse, you can use the cells of those mouse to get their genomic DNA, and you can analyze the presence of that gene of interest which you have sent with the help of a PCR. And this is the time when you came to know whether you have successfully incorporated the external piece of DNA or not. And there are a lot of troubles associated that you don't know where that external piece will be incorporated. हमें नहीं पता कि वो कहाँ जाएगा. It can go, but अगर वो चला गया है, you might be able to know after that. Not at that time when you have done that experiment. This experiment was done with the help of an holding pipette, a pipette which is used to hold the zygote. Zygote is comparatively bigger cell, so you can hold it. Usually it was held within the groove of that pipette here. But a very wonderful science, अच्छा साइंसदान वो है जो इसको pipette के बाहर भी help कर सकता है. Because the pressure sensors and all these things, you need to be a wonderful artist in order to do that experiment. Hold the zygote with one pipette using the suction here. And then you can see this one. This is the syringe injection needle. It's a very small injection needle. And that injection needle comes from a source, and that source contains those things. Those things might be viral vectors, virus particles, or plasmids. And you inject it right within the zygote. Assuming what? You are assuming there are nucleus of the zygote, and that nucleus contains chromosomes. And you know there are two chromosomes, now a pair of chromosomes there. Half of the chromosomes comes from the sperm, the other half are already present here. So you are sending this thing right within the cells, assuming that those particles will be able to penetrate the nucleus, and then they will send the external DNA. It will become incorporated into the genome. The rate of success is very, it's very less. So that is why it's a very time-taking and painstaking type of experiments. You inject a lot of zygotes, and you have no idea that you have successfully incorporated or not. And after like 4 weeks or 5 weeks, you came to know nothing happens. All of your, incorporations were failed at that time. So that is the limitation factor of sending genes right within the zygote, because we have to send it at a single cell stage, at a zygote stage. Ji Aisha, आपने हाथ खड़ा किया.
Sir, can the cell get damaged through when we're using an injection?
Yes, there are a lot of side effects. I mean, we should raise our eyebrows. Let's see. Let me create the eyebrows, raised eyebrows. Okay. We should raise our eyebrows for this process. ये क्या चल रहा है? What's going on? I mean, this is like manipulating the very sensitive and delicate thing. So whatever you are sending, it can go to some important locations. ऐसा भी तो हो सकता है ना? Whatever the things you are sending, it might landed in some important places. This is called off-site incorporation. Off-site things. So you are sending an important thing, and that landed at important places, destroying the function of certain other important things. This is called off-site integration, or I must say, and that leads to trouble. And those troubles are called as cytotoxicity. It causes trouble. But that is the thing you should keep in your mind. यार ये जो हम कर रहे हैं, that is not normal. That is not. All these things are contributing in decreasing the success rate of such experiment. That is why जो लोग ये काम करते हैं वो skeleton बन जाते हैं इस तरह का. They are the people who are always on the weight, and they came to know about their troubles. Oh my God, failed experiment because of the microinjection 4 weeks ago. And they suddenly came to know that out of all and everything, they failed to get the products. There are different, so if we want to increase the efficiency, we should find wonderful methods of delivering this external gene. So we need to create delivery systems. Now we are pro. Now we are very much expert in sending the delivery system. If you remember the Pfizer vaccine, sending the RNA to the humans, sending the RNA to the human cells, and we have created a delivery system which packaged the viral RNA, part of the viral RNA, and that is able to deliver the material to the human cells. And it's amazing. I mean, human cells are not laboratory. It's not a laboratory conditions. So Pfizer vaccine is a wonderful miracle, I can tell you. It's sending something right within the cells outside the laboratory. It's amazing. So we need to develop the delivery system. So retroviruses are used, as I speak earlier. They are used to deliver material which can be incorporated into the gene. If you have a better delivery system, the success rate will be better. Things can be done before the fertilization as well. People can do the manipulation in the sperms. If you want, you can make changes in the sperm, or you can make changes in the egg, and then allow those modified eggs or modified sperms to go further to create a zygote. This can also be done. People are doing wonderful things to find better ways and better delivery system. I mean, look at that. I mean, we develop certain antibodies which can bind themselves with the sperms, and then we use the sperms as a delivery system. You know, sperms know where to go. Sperms know where to go. Where to go? Sperms need to go. They need to swim towards the egg, because the eggs are releasing certain chemicals, and those chemicals attracts the sperms. And you can use the sperm as a delivery system to send material to the egg, and the other things as well. Okay, we will study